Okay. Assalamu alaikum, everyone. My name is Rumaan Ahmed. I'm a second year medical student, and today's class session is going to be on insulin, glucagon, and diabetes mellitus. Okay. So I already know what you're all probably thinking right now, which is I've already done this before, right? This was already. So I already know what you're thinking about. Like we've already done this in more. Why are we doing this again, right? So, like basically like this, okay? So you've watched this part before. There were two pals on Mo about this already. One was on insulin and glucagon, and then one was on diabetes. So I made a specific slide just to show like how this differs between ERP and Mo. Okay, so here you have mole and ERP. This is where they clash, basically. This is where all the information meets up. So this is the similar information. I went over the file sessions in mole as well. So there's a couple of things that are going to be repeated. But like there are some things that are mentioned ERP specifically. There are some things that are mentioned in mole. So yeah, you have to study them separately if you want to get an A in both, inshallah. Okay. So without further ado, let's begin. So this is the table of contents. Okay. So we'll study histology and physiology, insulin versus glucagon, diabetes mellitus, where we'll discuss type 1 and 2, and then we'll compare between the types, okay? So first off, we'll start with the basics, which is histology, okay? So you've already taken the pancreas as an exocrine gland, basically, in GIT, where they release their digestive juices, okay? They go into the duodenum, which is a C-shaped organ, basically, okay? Which was shown earlier on, okay? Um, but this block discusses the endocrine functions. So in ERP, you're going to be studying about the islets of Langerhans and the two main hormones that are released from the islets of Langerhans, which are insulin and glucagon. Okay. One main thing that I've seen questions about is this one bullet point here. Okay. So there are 1 to 2 million islets of Langerhans present in the pancreas. Okay. So they can ask you which of the following statements is true, and then this can be one of the options, and this is the correct option. Okay. The acini is basically the duct system. So this is the digestive juices. This is what you studied in GIT, and this is the stuff we're going to cover in today's part. Okay. Usually they don't ask about size, so don't worry about this bullet point, but it's added for completion's sake. Okay, so this is what I was talking about. So this is the duodenum. This is where the digestive juices are added. But it's like when we're talking about the endocrine portion, we're talking about blood vessels because hormones are released into the blood. They are transported to a target organ where they act and function based on a receptor, which can be on the cell surface membrane. It can be within the cell. That's covered in Hormones 1 and 2 by Dr. Simon. Okay. So this is a histological picture. This shows you the acini and the islets of Langerhans. So islets are basically like islands, and they're surrounded by this acini. Okay, so this is like the water. This is the land. In the middle, you have the beta cells. Surrounding it, you have alpha cells. Okay, so those are like two of the three main principal cells that you need to know, which are mentioned here. Okay, so you have the acini again here. You have the islets of Langerhans here. The alpha cells are in the periphery. Okay, so you see them here. The beta cells are near the middle, and the delta cells are somewhere in between. So between the periphery and the middle. The beta cells, they form most of the islets of Langerhans, so 60% of the total. The alpha cells form 25%, delta forms 10. Beta cells secrete insulin, alpha cells secrete glucagon, delta cells secrete somatostatin. So you need to know these three. Okay, these are the main ones. Um, Compared to last year, your slides were like there were 6 slides that were removed, and most of them were histology. So this is a summary table basically, so you can go over it. A cell type A is basically alpha cells. Those are for glucagon. They're 15 to 20%. Mallory Azan is a way of staining all three of these cell types together so that you can compare. So alpha is red, beta is brownish orange, and delta is blue. Okay, beta, as we said, is the majority and it secretes insulin, which makes sense because as we'll study later on, okay, insulin has a specific function which most other hormones can't do. Okay. Uh, so about the granules, you don't really have to know these in detail, I think. Okay, but it's okay. It's better to know. So go over the details. But again, size, they don't ask about this. This is more important. Okay. All right. This is what they removed from the slides. So uh, he removed these, but like this can be an option, so like a throw off option. So PP is basically pancreatic polypeptide cells. These are stimulated gastric leaf cells. So like most of this is related to GI, so they removed this. Okay, this is more relevant to your topics in ERP. Okay. Secretin acts locally to stimulate bicarbonate motilin. Okay, that's for gastric and intestinal motility. So like most of them, they like follow the name. Their function follows the name, so it makes sense. Substance P, you'll take this in neuro. It has neurotransmitter properties. This is for pain, basically. Yeah, you don't need to know most of these. Okay, just like go over them if you want to. So FYI. Again, this is another picture. This is really good. This is something that they can ask you about in OSPI. So again, beta cells in the center, okay, in the middle, and according to Mallory as in staining, they should be brownish orange. So this is correct, okay. Alpha cells in the periphery, they should be red, so that's good too, okay. And then delta should be blue, just like over here, they aren't, okay. Um, and then you have the capillaries, of course, because these hormones need to be secreted into them. One more thing, yeah, the percentages here are relatively like a good representation. So beta is the most, about 60 to 70, alpha is about 20, okay, and delta, which secretes somatostatin, is about 5 to 10. Okay. So here's a question for you guys to practice. The parents of an 8-year-old boy are concerned about his weight loss following a flu-like illness. Urinalysis reveals glucosuria, glucose in the urine. Okay, his fasting blood glucose is 220 milligram per deciliter. Normal is 50 to 100. So his fasting blood glucose is high. The patient child is diagnosed with diabetes mellitus type 1, which we'll discuss later. It's like which of the following cells in the endocrine pancreas produces insulin? So I'll give you like about 30 seconds because this is a simple one. So you can pause the video if you want, just like that. Okay, I think that's enough. Yeah. So the answer is, of course, B, beta cells, okay? Alpha cells, again, glucagon. Delta cells for somatostatin. F cells and PP cells, like I said, they are more like throwaway options because they aren't going to ask about it. But this was for pancreatic polypeptide, okay? You can go back if you want to refer to any of this, okay? To the table. So this is where the main lesson starts. This is where most important parts begin. So insulin as a structure, so insulin has a molecular weight of 5808, okay? So 5808. You don't really need to remember this, but it's important for comparison. So I'll explain why in a minute. Is a polypeptide made up of an alpha chain and a beta chain? So two chains, okay? And there are three disulfide bonds in one molecule of insulin. So there's one intramolecular, which means within the molecule, okay? And I'll show it to you in a second. And two intermolecular, which means between two chains, okay? So intramolecular within the chain, two intermolecular between the chains. And when the two chains are split apart, their functional activity is lost. So I'll show it to you on the structure, okay? So this is the general structure of proinsulin, not insulin, okay? So insulin would not have this part right here, this white area. Okay, this is a connecting peptide or C peptide, and I'll tell you what it is in a second. This is an alpha chain, a beta chain. There are three disulfide bridges you see here. Okay. Yeah, so that's basically it. This is the intramolecular bridge. Okay, this one right here. And the intermolecular ones are between the two chains. So alpha and beta, they're connected by two intermolecular ones. Okay. Of course, the disulfide bridges are between cysteine molecules. So this is from mole, I think, and F and D maybe. So you should know this, okay? So if they ask you about which is interesting. If they ask you about how many cysteine molecules are there in a molecule of insulin, say 6 because yeah, 3 disulfide bridges and 2 cysteine molecules per bridge. So yeah, that's mostly it for this structure. Okay, so you know, of course, you don't need to know the amino acid sequence. Yeah, just know there's 2 chains, there's 3 disulfide bridges, 2 intermolecular, 1 intramolecular. Okay. So insulin, how is it synthesized? Okay, so insulin is initially synthesized as something called a pre-pro hormone. Okay, so this pre-pro hormone will contain a signaling peptide, a connecting peptide, and the insulin molecule itself. Okay, what we saw here is a pro hormone. We'll see what it means in a second. Okay, so first off, when insulin is synthesized as a molecule, okay, the signaling peptide will be cleaved off in the something called the endoplasmic reticulum. You should be familiar with this by now. And then the remaining molecule will be called proinsulin, which we saw earlier. Okay. Then the C peptide or the connecting peptide is cleaved off in the Golgi apparatus, and you're left with insulin. It's really, really important to note that this C peptide is always produced and secreted in equimolar amounts with insulin in secretory vesicles. So let me explain what I mean in a sec. Okay, here's a diagram. So you have mRNA here, okay, that's synthesized. That mRNA will be translated by these ribosomes on rough endoplasmic reticulums, okay? This translated chain of amino acids, okay, it'll form 2 chains, an alpha chain and a beta chain, which will be connected by the 2 intermolecular disulfide bridges we talked about, okay? Then there's also going to be the C peptide and a signaling peptide with it, okay? So that's why that will be called a pre-pro hormone. The function of the signaling peptide is to direct this large pre-pro hormone into one of the endoplasmic reticulums. So you took this in more with Dr. Abdul Jabbar. It's better to go back to the slides if you want to revise what a signaling peptide does. But basically, the molecule is going to be synthesized out here. Okay, the signaling peptide will take it into the endoplasmic reticulum. Okay, over there, now the signaling peptide has no more function, so it's going to be cleaved up. Now you're going to be left with proinsulin, which is this molecule right here. But you don't need the C peptide either, okay? You just need this whole part for insulin, okay? So how do you cleave off the C peptide? You're going to take it to the Golgi apparatus here, okay? You're going to cleave off the C peptide, and then you're going to get insulin and C peptide in one vesicle, which looks something like this, okay? So this is what you'll have as proinsulin, okay? When this is cleaved off, you'll have insulin here and the C-peptide here. The importance of the C-peptide is that it's always produced in the same amounts as insulin. And it makes sense because whenever you're translating that insulin strand, you're going to have this with it always. Why is it important? I'll get to it in a second. But like, remember, C-peptide and insulin are always in the same vesicle if it's endogenous, okay, which means it's synthesized within the body. So measurement of C-peptide by radioimmunoassays or scans and stuff can be used in insulin-treated diabetic patients to determine the natural insulin production levels. So how much insulin is produced within the body can be determined by the C-peptide levels. So even if because you can't tell if the insulin you're measuring is the insulin that's produced within the body or it's the one that's been injected into the patient. So the only way you can measure it is by this C-peptide. It's basically like this will tell you that this one is secreted by this one is produced within the body. So whatever amount of C-peptide there is, that's the amount of endogenous insulin, basically. And patients with type 1 diabetes usually have greatly decreased levels of C-peptide, again, because they have low levels of insulin that's synthesized within. But we'll discuss diabetes in a second. But remember C-peptide, okay? Insulin on the move. So half-life reference. Insulin circulates in the body, blood unbound, because it's a hydrophilic molecule, okay? So it does not need to bind to a protein to have, to circulate within the body, basically. That's for lipophilic ones. Lipophilic does have the advantage of going straight through a cell membrane because there's nothing that's inhibiting it. Hydrophilic ones, like insulin, require a receptor on the cell surface membrane. So this was covered in hormones 1 and 2, but like just a quick revision. So insulin circulates in the blood unbound. It has a half-life of 6 minutes, and it's cleared from the circulation by 10 to 15 minutes, okay? So endogenous insulin, it doesn't stick around that long. Except for the portion of the insulin that combines with the receptor in the target cells. So whatever binds to the receptor, and you took this in cell signaling in mole, the receptor for insulin is called tyrosine kinase. Okay, whatever binds to it, it's going to remain. But like the rest of the insulin molecule, it's going to be degraded. And there's an enzyme which is conveniently called insulinase. Okay, and that's mainly in the liver, and to a lesser extent in the kidneys, muscles, and slightly in most other tissues. Rapid plasma removal. So it's really important to control the levels of insulin because, as you'll see later, you've covered this in high school. So the basic function of insulin is to decrease the blood glucose levels. But you don't want to decrease it too much because you can have something called a hypoglycemic shock, which we'll mention later on. You don't want it to like be completely off because if you have hyperglycemia, that's bad too. So it needs to be tightly regulated. So that's important. Okay. Okay, insulin secretion. So first of all, we mentioned insulin is produced in the beta cells, okay? Whenever the blood glucose levels are high, okay? So whenever there's not necessarily, okay, sure, hyperglycemia, okay, but we usually use that term for pathology, for example, diabetes. But whenever blood glucose levels are high, for example, after a meal, okay, this blood glucose will enter beta cells in the pancreas through GLUT2 transporter proteins at a rate proportional to blood glucose concentration. So basically, however much blood glucose is present in the blood, that's the amount, like a proportional amount, or basically an equal amount will go into the beta cells through GLUT2 transporters. There are different glue transporters. You must have studied them earlier. That's like to remember which one it is. At least this helps me. Insulin is produced in beta cells. So B is the second letter of the alphabet. So GLUT2, if that helps in any way. GLUT2 transport proteins. Okay. So within the beta cells, whatever glucose enters is going to be phosphorylated. And this is a rate limiting step. And the importance of the phosphorylation, sure, it helps later on because you're going to do glycolysis, oxidative phosphorylation, everything respiration, which you studied in the first semester in mole. But like the main reason you're phosphorylating it is now the glucose can't leave because for glucose you have these transporters like GLUT and other transporters. But like for glucose 6-phosphate, for example, you don't have a specific transporter, so it's going to be trapped within the beta cell. Now it has to be converted to ATP, which is the next step. That ATP will inhibit something called an ATP sensitive potassium channel. Okay. And this potassium channel is normally involved in efflux of potassium, which is basically potassium ions moving out. If they don't move out, now potassium ions will basically like build up inside. That will cause a depolarization within the cell membrane, and this will open calcium ion channels. And these calcium ions will enter. They will cause vesicles, which already contain insulin, to move to the cell surface membrane, fuse, and exocytose the insulin. Okay. So that was a lot of words. Let's look at it that way. Okay, you have the GLUT2 transport here. You're looking at a beta cell. So glucose enters from the blood in a level proportional to the amount of blood that glucose that's outside. Okay, this glucose enters. Glucose 6-phosphate is formed by glucokinase. Okay, so again, mole. This glucokinase forms glucose 6-phosphate, which will be converted to ATP through a series of reactions. So we say oxidation. Okay. ATP is formed. High ATP will inhibit this channel right here. It's called an ATP sensitive potassium channel. Okay, this will prevent potassium ions from going outside. Okay, so basically potassium ions will build up in here. There's going to be depolarization. Then voltage gated calcium ion channels will open here. Calcium ions enter. Increased intracellular calcium will cause vesicular release. So this is kind of similar to, if you look at just this part here, like this section right here, it's kind of similar to what you see in nerve action potentials. So like at the synapse, when depolarization occurs, calcium ions enter, okay? They go to vesicles, they cause release through exocytosis. And this insulin will again go to the blood vessel. This will, go to its target organ, have its effect. Okay. All right. Same thing. Let's going over it again, just so that it's like really clear. Glucose from the blood will go in to a beta cell through a GLUT2 transporter protein. Okay, this glucose will be converted to glucose 6-phosphate by something called glucokinase. Okay, this glucose 6-phosphate will be converted to ATP through a process called oxidation. So, Krebs cycle, oxidative phosphorylation, everything we studied in more. This ATP will inhibit something called an ATP-sensitive potassium channel. Once this is closed, this will cause depolarization because you see potassium ions are building up. Potassium ions building up will cause a depolarization, which will activate this calcium ion channel. So this voltage-gated calcium channel will open up, calcium ions enter, they cause these vesicles to really release, okay? So there's going to be fusion, there's going to be exocytosis, okay? Okay. Here we have a question. So researchers studying the pathophysiology of diabetes mellitus identify a patient with an inherited mutation in the insulin gene. The mutation results in a single amino acid substitution in the proinsulin molecule. However, affected family members are asymptomatic and mostly have completely normal blood glucose levels. Analysis of the mature insulin molecule shows that it's identical to normal human insulin. The researchers conclude that the amino acid substitution affects a segment of proinsulin removed during post-translational processing of the protein. The polypeptide segment contains the mutation, which is most likely to undergo which of the following processes within pancreatic beta cells. So you have options here, A, B, C, D, E. Okay, so I'll give you about a minute. Yeah, sure, a minute. A lot of P's. Okay. Okay, so that's good enough. Okay, so the answer is E. All right. So let's go over the question, okay? So the question asked, researchers studying the pathophysiology of diabetes. All of this is fine, okay? But the main thing that they're asking, read the last statement. The polypeptide segment containing the mutation is most likely to undergo which of the following processes within pancreatic beta cells, okay? They're asking about something, they're asking about a single amino acid substitution in the proinsulin molecule, but it's not in the actual insulin molecule, okay? So something that's component of the proinsulin molecule, but not, it's not the insulin molecule itself, okay? So we're talking about the C-peptide, and we said the C-peptide is always stored in the same secretory granules right here. So that's why the answer is E, okay? Direct translocation across the plasma membrane is wrong. C would be for, okay, yes, endoplasmic reticulum, that would be signaling peptide. That's wrong because that's not a part of proinsulin. That's a part of pre-prohormone insulin. Degradation in proteasomes and lysosomes is wrong because we talk about lysosomes or proteasomes. So lysosomes are hydrolytic enzymes, proteasomes are for ubiquitin and the protein degradation pathway. So we didn't take that, okay? Next question. A 4-week-old infant is evaluated for frequent episodes of hypoglycemia. The patient is found to have elevated insulin levels during periods of fasting. Several members of the family had a similar condition. Genetic testing reveals a mutation involving potassium channels expressed in pancreatic beta cells. The mutated channels have a higher sensitivity to the substance that normally modulates their activity, causing increased beta cell secretory activity. Which of the following substances normally binds to and regulates the ion channels that are responsible for this patient's hypoglycemia? I'll give 20 seconds because there's just the answers. You don't have to think too much about it. Okay, so the answer is ATP. Okay, so because we said something that binds to and stops the potassium action, right? The ion channels that are responsible for the patient's hypoglycemia. So hypoglycemia, we said insulin. Okay, so whatever regulates these ion channels. Yeah. Okay, further mechanisms of insulin secretion. So other than what we talked about, we said glucose was the main key substrate for the release of insulin, right? Because we said it enters to the GLUT2 transporters and it causes effect. But you can have other nutrients and amino acids, which may also be used to produce intracellular ATP and stimulate insulin secretion. Because you studied this in mole as well when you're having the process of respiration, right? You can have multiple substrate. Glucose is the preferred one, but you can have amino acids, you can have fatty acids, anything else. Okay, so yeah, all of them can stimulate insulin secretion. Other hormones may increase intracellular calcium levels through other signaling pathways and enhance glucose effects. So glucose is entering, it's causing the ATP formation, it's inhibiting the potassium channel, which will cause the depolarization, increasing the calcium ion influx, and that causes the vesicles to be released. Sure, but there are other hormones that act on this calcium ion channel, which was the last step we said where calcium ions enter the cell and then they cause the vesicular release. So those can be glucagon, glucose-dependent insulin-entropic peptide, and acetylcholine. Okay, don't worry about too many of them. Just like get a general idea of them. I'll tell you how to memorize them later on. Other hormones which inhibit the exocytosis of insulin. So when you don't want insulin to be released, norepinephrine, alpha adrenergic. So remember this. This will come back in a second. And somatostatin. Okay. Somatostatin, which we said was from D cells. Okay. Pharmacological effects on insulin secretion. So, pharmacological, for those who don't know, basically is for drugs. Okay. So, what drugs can affect insulin secretion? Sulfonylureas. Okay. You'll study this in BOD in detail. But sulfonylureas exert their effects by binding to and blocking the activity of ATP-sensitive potassium channels, which will increase the insulin secretion. So it's basically doing the exact same thing that ATP is doing. So it's still going to increase insulin secretion because if it still binds, it blocks the potassium channels. You'll still have the calcium ion influx because potassium ion efflux is blocked. Okay, so you'll still have the vesicular release, and this is important in patients with type 2 diabetes mellitus. So I know I keep mentioning diabetes, and I say it's going to come back later, but it's like when it does come, there's going to be a lot of first aid tables and pictures and stuff. That's when you refer back to these slides. Okay. Okay, things to know from the table. I didn't show any table. What table am I talking about? This one. It's really, really, really big table. Okay. There's a lot of information that's mentioned here. I summarize it into 5 major points. Okay. So insulin is an anabolic hormone. Anabolic basically means build. So I know you took 6 B's for thyroid. So for insulin, you have 3 B's. Okay. So one of them is insulin builds. Okay. So the second one was insulin is secreted by beta cells. And the third one, as you'll see here, this is in the formatting error, it's for you to remember. Beta adrenergic will stimulate insulin secretion. Okay, so these are the 3 B's that I want. So insulin is an anabolic hormone, whereas glucagon is catabolic. So what I mean is see all these things that increase insulin secretion, increase blood glucose, increase blood free fatty acids, increase blood amino acids. They're like basically building components, building blocks. All of these can stimulate insulin secretion because all insulin wants to do is take all of these. It wants to make something better, big out of them. So, for example, from amino acids, it'll form proteins. From free fatty acids, it'll form triglycerides or fats, and then it'll deposit those in adipose tissue. From glucose, it'll form glycogen, and from the excess glucose, excess carbs that you can't digest, it'll form fats again, which will be sent to the adipose tissue. So, and as he mentions later on, as Dr. Joaquin mentions later on, Insulin is a hormone which is like whenever you have excess, whenever you have like available stuff, this hormone's going to be released because it's going to want to store everything. So yeah, beta-adrenergic and acetylcholine stimulation. So like we mentioned earlier, so acetylcholine, like it's for parasympathetic stimulation. Okay, this will increase insulin secretion because and this makes sense when you're resting, when you're like all calm, you've had a meal, you're not using up too much energy or anything. You'd rather have all of your glucose, etc., be stored so that you can use it in a later time rather than using it at that time because you're not, performing any movements, doing anything specific which will require all this ATP and glucose free floating in the blood, causing hyperglycemia. So yeah, parasympathetic will increase insulin secretion. Beta-adrenergic will increase insulin secretion. Alpha-adrenergic will decrease insulin secretion, which we mentioned here. Okay, so when I said norepinephrine here, that was alpha-adrenergic that will inhibit exocytosis of insulin. Okay, leptin is just something, I think you took this in GIT, something related to satiety. So that will also decrease insulin secretion. Obesity and insulin resistance, for example, in type 2 diabetes, will stimulate insulin secretion. Excuse me. And somatostatin and fasting will inhibit because when you're fasting, you want glucose to be sent outside because you already have hypoglycemia. Insulin will just make it worse. And somatostatin, like we said, from T cells, it's going to inhibit insulin secretion. Obesity and insulin resistance, why would they stimulate? We'll talk about type 2 diabetes later on. Because when you do have insulin resistance, basically insulin's unable to do its thing. It can't cause the hypoglycemia. So what your beta cells will still recognize that glucose coming in through the GLUT2 transporters. It'll still continue to have the exocytosis of insulin. So it's going to still produce more and more insulin. And we'll see the production of insulin, the release of insulin, its action, all of that later on in this part, inshallah. So yeah. That's most of the table. Um, a couple of things I didn't mention. This one right here, okay, gastrointestinal hormones, for example, gastrin, cholecystokinin, secretin, gastric inhibitory peptide, they also increase insulin secretion. Just like no in general, GI hormones, they increase insulin secretion. Glucagon, growth hormone, cortisol, sulfonylurea drugs, they increase insulin secretion. Okay, the rest was mentioned earlier. Okay. All right, insulin highs and lows. So when do you want to increase insulin? At what point does your body decide, okay, now the blood glucose levels are really high. I do need to secrete insulin to like maintain them, regulate them. So at the normal fasting level of blood glucose, which is about 80 to 90 milligrams per 100 milliliter, the rate of insulin secretion is minimal. So you don't really secrete too much insulin at that point. But it's like if the blood glucose concentration is suddenly increased to a level 2 to 3 times the normal amount, and I'll show you a graph which shows this, okay? And kept at this high level, insulin secretion increases markedly in 2 stages. Why two stages? I have an analogy for you as well, because like I'll show it to you in this graph right here. So first off, when you have an acute elevation of glucose, for example, like we said, 2 to 3 times the normal amount, the plasma insulin concentration will increase by 10 times within 3 to 5 minutes, which is which is a really huge, acute elevation in a really short time. And as a result, this is as a result of preformed insulin being secreted. So just assume your beta cells have a stock of insulin basically. So they have all this insulin stored and they're just waiting to be exocytosed. Okay. So whenever the blood glucose concentration it increases to like 2 to 3 times its normal amount, all of this insulin is going to be exocytosed. Okay. Because again, that blood glucose is recognized by GLUT2, all that. Like keep revising. Okay. That's how you're going to remember this. Try to link a later concept to a previous one. So okay, that preformed insulin secrete as your stock, it's slowly like starting to thin out basically because you're releasing all that insulin, but you don't have an unlimited supply of insulin. And even if you did, you keep releasing that all that insulin. We did say that insulin has a specific half-life. After that, can't keep releasing it. So you need to form more insulin, and that's why you have this second rise right here. So this is the first rise. This is because preformed insulin, all the insulin that was already within the beta cells waiting to be excreted, was released. Okay, and that's why your plasma insulin levels go up high all the way here. After that, it drops because you're out of insulin for now. Okay, you're still going to secrete more insulin, but you need to form new insulin, which is the second point here. Okay, so the rise in the plateau is because some of the preformed insulin is also being released, and then there's new insulin formation by the activated enzyme system, and that new insulin formation would include the pre-pro hormone, then that's cleaved to form the proinsulin, then you'll have the insulin. Okay. So that starts at approximately like 15 minutes, which is shown right here, okay? And then insulin secretion rises, goes to a plateau, okay? One way you can remember this, like I was saying, stocks, right? So let's say you have a specific stock of something that you want, okay? You go to the grocery store one day, you buy a whole stock of, for example, let's say jam, okay? You get a lot of jam one day, take it home, plus you're done. The next day you go to the grocery store, plus they're all out of stock. They don't have any more, right? And you're gonna have to wait a bit because they're gonna have to produce more jam. Okay, so that's basically this wait time right here. Okay, and they can't have like an unlimited stock because there's like it's maybe an expiry or something. Okay, and that's supposed to be like an analogy to remember that insulin also has a half-life. Okay. Okay. This is it about insulin and its preformed part and the new insulin that's going to be formed. Okay. So what we said earlier about insulin rising to a level 2 to 3 times its normal amount, I'll show it to you here. So let's say plasma glucose concentration went 2 to 3 times its normal amount. So it was 100 here. Okay. So this is minimal secretion, which we said was fasting. Like look here, fasting level of blood glucose 80 to 90 milligram or 100 milliliter per 100 milliliter. The rate of insulin secretion is minimal, which is true here. So you can see from the graph. But then if you increase the blood glucose concentration to 200 or 300, now your blood insulin secretion levels will be really high. So that's if you plot up from the x-axis and then you match it to a y-axis, it's going to be 10 times normal, which is true, which we did say here. So like this graph basically proves it. You know? Okay, so this is basically how insulin acts on a receptor. So this is insulin's receptor, okay? Like we said, it's tyrosine kinase, again, mole, okay? So this is cell signaling. You can revise the function of tyrosine kinase, the IRS, this is insulin receptor substrate, not the other one. And phosphorylation of enzymes, which causes protein synthesis, fat synthesis, all of this. So first off, insulin binds to this alpha subunit right here, okay? This is basically the extracellular domain, the receptor, but this is part of the tyrosine kinase. You know how the tyrosine kinase works? There's autophosphorylation. So this one phosphorylates this one phosphorylates this. You have an IRS. There's a cascade of events, which is more relevant to mold, so I'm not going to cover it now. But there's protein synthesis, fat synthesis, glycogen synthesis. As you can see, synthesis, so it's an anabolic hormone. It will build, it won't break down, okay? There's growth and gene expression, and growth we'll cover in a second because growth is related to growth hormone, okay? And it has a synergistic effect. We'll talk about that in a second. And then another one is glucose transport, okay? And this is one of the parts of insulin's action, okay? It's going to cause these vesicles to move to the membrane so that more glucose can enter, which makes sense. You want to decrease blood glucose outside, so you want to increase the concentration of these glucose transporters, so all of this blood glucose can enter into the cell. Once the cells take it up, your extracellular glucose will decrease. Okay. This is a very complicated diagram I found on Google, but the only reason I added it is because I thought this physical looked really, it was much better than here. Okay, so this is more of what it looks like basically, okay? So insulin binds its receptor, tyrosine kinase, phosphorylation, IRS, again, insulin receptor substrates, PI3K, phosphatidylinositol phosphate, whatever the kinase, okay? and then it phosphorylates something. All of these, you don't need to know these, okay? But you do need to know this: protein synthesis, glycogen synthesis. There's going to be lipid synthesis as well, so you're going to form triglycerides. This AKT is going to cause these GLUT4 to be translocated, okay? So that's basically the transport for other cells excluding beta cells. So for beta, we said it was GLUT2 for general cells, which will be involved in the uptake of glucose due to the insulin action. That's going to be GLUT4, okay? So yeah, these vesicles come in here, they'll fuse with the membrane, and because these GLUT transporters, they're part of this phospholipid bilayer, they're going to be like a transmembrane channel, basically. So they're going to allow glucose and glucose molecules to enter. Okay. Yep, that's basically it from here. Okay. So this is the action of insulin. Insulin acts on tyrosine kinase receptors, like we mentioned. They follow a cascade of events, and GLUT4 transporters fuse with the membrane, increasing glucose influx. So we talked about all of this here. But like, what about what happens after the glucose enters? Glucose is converted to glycogen in the liver and muscle. Okay, this is important. Remember this. So it's converted to glycogen in the liver and muscle, and insulin will also inhibit glycogenolysis. We'll talk about the specific terms and what they mean, but like glycogen and lysis is breakdown. So hydrolysis is breakdown by water. Glycogenolysis is breakdown of glycogen. So you're going to inhibit this, so you'll have more of these stores, more of glycogen. Insulin inhibits gluconeogenesis, okay, which is glucose. Neo is new, genesis is generation. So generation of new glucose or production of new glucose. So I know it's anabolic. Yeah, insulin is anabolic. Gluconeogenesis, even though it sounds like it's anabolic, it's catabolic. This is something that glucagon does. But it's not specific to glucagon. Glucagon is more involved with other processes. We'll talk about this. But mainly for glucagon, it's going to be glycogenolysis. Okay, gluconeogenesis is for epinephrine and the others. Insulin inhibits gluconeogenesis by increasing the activity of fructose 2,6-bisphosphate, which increases plasma fructokinase activity. These names sound familiar to you because they're from Mo. Okay, so these are Dr. Hanad's styles from last year, and I brought them in just to like emphasize on this because he didn't emphasize on this part, but it's important to know because it's part of insulin's mechanism of action. So there's two pathways that it can go. Okay, so one is fructose 6-phosphate to fructose 1,6-bisphosphate, and if you remember your mole one, this is in the direction of glycolysis. Somewhere along this line, you have pyruvic acid. Okay, and then pyruvic acid will be the ATP that we talked about earlier. Thus, if you go backwards from fructose 1,6-bisphosphate to fructose 6-phosphate, then you go to glucose 6-phosphate, then glucose. That's gluconeogenesis. So this way is glycolysis, this way is gluconeogenesis. You want to inhibit this way, or you want to do more of this basically, right? So on or off switch basically, right? So glycolysis increase is equal to glycolysis on, decrease is no glycolysis. Yeah, so basically the opposites, right? Okay, so you're increasing phosphofructokinase activity, you're increasing the activity of this enzyme here, so you're going to cause glycolysis basically. And here it is again. Okay, I added this slide just because it mentioned insulin and glucagon. So levels of fructose 2,6-bisphosphate rise with high insulin. When you are fed, when you're well fed, you had a meal, you have a lot of glucose, amino acids, free fatty acids floating around. You can store them. You want anabolic reactions to happen. You want to store all of this energy for later on. So there you use insulin. When you're fasting, as in this month. So yeah, you have glucagon, which is high, okay, and this is going to increase gluconeogenesis, glycogenolysis, all of that stuff, okay. So it drives glycolysis. So glucagon wants gluconeogenesis, insulin wants glycolysis. Okay. Glucose transport and glycogen synthesis and storage. Okay, so this is one huge picture from first aid. Dr. Yakin added a screenshot of this, and it's going to be covered in the next few slides, so don't worry about it. There are a lot of points that are mentioned here, but I want to cover a lot of them separately because they'll come again in diabetes. So there's going to be an abnormality. So basically, these are the effects of insulin. If you have a problem with insulin, none of these will happen. So all of these arrows will go opposite. So there's decreased glucose transfer, decreased glucose synthesis. We'll cover all of them later. So this is important. Insulin is a hormone associated with energy abundance. So again, it's an anabolic hormone. Do not forget that. Excess carbohydrates are either stored as glycogen, mainly in the liver and muscle, or stored under the stimulus of insulin as fats in adipose tissue. Okay? So, like I said, carbohydrates will be converted to glycogen. But if you have too many carbohydrates where you don't want to convert them to glycogen, you'll start converting them to under the stimulus of insulin as fats in adipose tissue. That's why people who take too much carbohydrates will, end up having more adipose tissue. Yeah, that's a way to put it. Okay, so this is very important. Very important. Like one of the most high yield things from the both lectures together is the cellular uptake of potassium ions and amino acids and how insulin is relevant to it. So insulin promotes potassium ion uptake into the cells at the same time it promotes glucose uptake. Okay, so potassium and glucose together, okay, try to associate them together. Potassium ion uptake is increased into the cells by increasing sodium potassium ATPase activity. You've been introduced and you've been familiar with this channel before, okay? So it's for active transport. Insulin increases this activity, so potassium ions enter more, okay? So yeah, you'll have more intracellular potassium, less extracellular potassium. So you'll have hypokalemia outside and uh, basically more potassium intracellularly. Yeah. And this is good if, for example, the patient is suffering from hyperkalemia, then insulin can be a good, standby as a treatment. I mean. This protects against an increase in serum potassium. For example, when you ingest potassium in a meal, it's taken up with glucose and other nutrients. So again, hyperkalemia. It also has an effect on hypothalamic satiety center independent glucose. He didn't really stress on this, so you don't really need to know. But this is covered in slide 69. Important for diabetic ketoacidosis. So in diabetic ketoacidosis, since you have uh, we'll study this later, but type 1 diabetes, you don't have insulin. Okay? You don't have insulin, you can't promote this potassium ion uptake. Okay? So you'll have potassium ions more outside. And we'll cover what happens because of that later on. Increased protein synthesis, again, insulin is anabolic. Okay? It's going to want to increase protein synthesis. Okay? And how does it do that? It increases the amount of amino acids that are taken up, basically, and it'll convert them into proteins. Okay? One important detail I'll mention right now, and I'll try to mention later on with gluconeogenesis as well, is that in gluconeogenesis, you also need amino acids, okay? You want to form glucose, so this is important, okay? So one, insulin has a direct effect in taking up amino acids, and two, that gluconeogenesis also requires amino acids. It also inhibits breakdown of proteins already present inside the cells. So someone with insulin deficiency will have both of these. He'll have a breakdown of proteins already present, and he'll also not take up amino acids. So amino acids will be free floating and breakdown of proteins present. So that's called muscle wasting. So mostly proteins are in the muscle, right? Excess nutrients are stored as proteins in the muscle. So muscle wasting, so that those patients will be really thin. They lack muscle bulk. Okay. So yeah, insulin binds to tyrosine kinase, causes phosphorylation, PI3K pathway. And then you have glycogen lipid protein synthesis because it's an anabolic hormone. It's going to cause these vesicles containing GLUT4 to be transported to the cell surface membrane where this GLUT4 can now reuptake glucose. Remember, this is another cell. This isn't beta cells, okay? So GLUT4 for normal cells, and then for beta cells, you have GLUT2. And then RAS-MAP kinase pathways for cell growth and DNA synthesis. So again, we'll talk about growth with growth hormone in a second. Decrease ketogenesis and lipolysis. So overall effects of insulin is to decrease fat mobilization and oxidation, instead increasing fat storage. So in people with insulin normally is supposed to be a storage molecule, right? So whenever you're taking in, like we said, too many carbs and stuff, it's going to be glycogen, and then the excess carbs are going to be stored as fat for storage. So insulin wants to produce fat, but it wants to prevent breakdown of fat. Insulin wants amino acids to be converted to proteins, but wants to inhibit breakdown of protein. Insulin wants glucose to be converted to glycogen, but wants to inhibit gluconeogenesis and glycogenolysis. So it just wants to keep everything stored. It doesn't want anything to be in the serum, you know? As a result, insulin decreases circulating levels of fatty acids and keto acids. Okay, because I think you'd study this in more fatty acid oxidation, but keto acids was covered later on in that lecture, where you had the beta hydroxybutyric acid and acetic acid formation. So it's good to revise that lecture when you're studying this keto acid part of this lecture, because it's important. Okay, so when you're preventing fatty acid oxidation and stuff, you want from keto acids. One characteristic feature of ketones present in a patient is they have a fruity smell in breath. So, you know, it smells sweet, basically. And it's going to be covered later in diabetic ketoacidosis. So yeah, again, this is important in diabetic ketoacidosis. As a result, insulin decreases circulating levels of fatty acids and keto acids. So decreasing so increased fat deposition and decreased lipolysis and keto acid formation in liver, because decreased lipolysis means less acetyl-CoA is available for keto acid formation. So please advise more, because acetyl-CoA is more relevant to more. I don't want to, dive into details of that. But if you have any questions, again, you can ask me anytime. It's fine. Okay. So this is a summary table of all of the actions of insulin we talked about. Okay, so it increases glucose uptake into the cells through GLUT4. It increases glycogen formation by doing the glucose 6-phosphate thing, and then the phosphoglucokinase and stuff is for inhibiting the gluconeogenesis pathway. It's for glycolysis, right? But like in general, you form glycogen from glucose after you take it out through the GLUT4 transporters. You decrease glycogenolysis, okay? You decrease gluconeogenesis, like I mentioned. It increases protein synthesis. Again, it's anabolic, okay? And you mentioned this a couple of times, so it's important to remember. Increases fat deposition, decreases lipolysis, because you want to inhibit fats from being broken down, but you want to increase the fat deposition. And increase potassium uptake into cell, and again, associate this with glucose. So whenever you're taking in glucose, you're also going to increase the potassium ion uptake into the cells. So that means hypokalemia. So serum levels of potassium will drop and intracellular potassium will increase. What happens to the blood? So again, let's relate this because we just finished it. So decreased blood potassium. Because you decrease lipolysis, no fatty acids produced, no acetyl-CoA, no fatty acid oxidation, no keto acid formation. Okay, no beta hydroxybutyrate and acetic acid, right? Since you increase fat deposition, you have decreased blood fatty acids because they're taken up for this fat to be formed, for the triglycerides to be formed. Decreased blood amino acids, so you can't have like gluconeogenesis. At the same time, you're also producing proteins from those amino acids. And the glycogen formed is from the glucose, so that's why your blood glucose levels will decrease. Okay, so insulin's effect on brain utilization of glucose? No. Okay, and yeah, because it doesn't have an effect on the brain utilization of glucose. The brain is special in the way that it uses only glucose for energy, and that's why it's not limited by insulin in any way. So even if you have hyperinsulinemia, even if you have a problem with production of insulin and you have hyperglycemia or whatever, okay, your brain cells are permeable to glucose independent of insulin's effects. So this is a good thing, because that means diabetic patients, they don't have like mental problems, basically, you know? Okay, so failure to maintain glucose levels above a certain threshold will lead to hypoglycemic shock. So this third point that I mentioned is important in the fact that, since it only uses glucose for energy, if glucose is depleted, you'll have hypoglycemia. If you have hypoglycemia, the brain's going to be affected. Okay. The manifestation of these problems are fainting, seizures, and coma. You'll take seizures again in neuro, so that's a year 2 thing. But just remember, okay, seizures, fainting, coma. So these are like brain-associated problems because it's not receiving the right amount of glucose. Okay, again, insulin's anabolic. Okay, so I'm repeating this again and again because I'm hoping this will stick if you like keep listening to it again and again. But please try to link the concepts and go back if you don't understand anything, or at least ask me. Like my contact information was added at the beginning of the slides. So if you have a nutrient, for example, glucose, okay, insulin will decrease it. If you have a nutrient like fatty acid, it'll decrease it. Keto acids, it'll decrease it. Amino acids, it'll decrease those two. So basically, any nutrient that can be built into something that can be stored, insulin will decrease it by taking it up and forming that stored molecule. So here you have glycogen. That glucose 6-phosphate can also be targeted towards the pyruvate pathway to form CO2. You have fatty acids and keto acids. Okay, you have glucose. Yeah, so to form fats here by sending those fatty acids to the adipose tissue. Excess carbohydrates in the form of glucose, they can be turned into fats as well, and they'll be stored in the adipose tissue as well. And amino acids, like we said, most of the muscle bulk will be proteins. So these amino acids will be sent to the muscle. Okay, and some glucose as well. Okay, insulin and growth. Okay, so sorry, one second. Okay, yeah. Insulin has a role in growth as it's involved in protein synthesis. And how did they figure this out? So in a depancreatized, so the pancreas were removed, hypophysemized. Okay, that's the hypophysis, pituitary gland, that's removed as well. Okay, rat has almost no growth without therapy. Now, in this rat, when they administered either the growth hormone or the insulin, there was barely any growth, like minimal. You see these plants right here. But when they're administered together over here, look at the 2 points, okay? There's a steep dramatic effect. There's a dramatic growth. So the conclusion was these two hormones work together to cause growth, basically. And the idea is maybe they like trigger the uptake of different sections of amino acids. For example, just random amino acids for the sake of like understanding, right? So growth hormone can cause the uptake of tryptophan, lysine, something else, maybe glycine or something. Okay, then insulin could be like phenylalanine, tryptophan, tyrosine, all the others. Okay, so different section of amino acids, but all of them are required for growth. If you have a lack of some amino acids, you can have growth, right? So you need both together. Okay, so yeah, that's important. Okay, next one. All right. One sec. Okay, so um, glycogenolysis, gluconeogenesis, ketogenesis, and lipolysis. All of these are decreased when you have insulin because, like I said, it's not catabolic, it is anabolic. The catabolic ones are glucagon and epinephrine. Okay, they are going to cause glycogenolysis, so breakdown of glycogen to form glucose. Another process that forms glucose is gluconeogenesis. This involves different substrates. So maybe I'll use fatty acids, amino acids or something. Okay, and I'll somehow form glucose. Okay, ketogenesis. So those fatty acids they can be oxidized. Okay, and then beta hydroxybutyrate and acetic acid. Again, please refer back to mol for this. Keto acids. Okay, and lipolysis. So breakdown of lipids. Okay, all of these are due to glucagon and epinephrine. One more interesting thing to note is that in addition to glucagon and epinephrine, you also have growth hormone and cortisol, which have similar effects to this, but you have nothing similar to insulin, which is also why it makes sense that you have beta cells as the highest percentage in the islets of Langerhans, because this is the only hormone that's causing all of these effects. And we'll see in diabetes, if there's a problem with this hormone, then there are going to be manifestations. We'll see them in a second. Okay. All right. So effects of other hormones on carbs and lipid metabolism. Okay, growth hormone, which is released from the anterior pituitary gland. So again, we're going to talk about other hormones that you've already taken previously. So growth hormone and cortisol. okay, which is from the adrenal cortex. Um, cortisol, just a refresher, this was ACTH, okay, and growth hormone releasing hormone, or stimulating the release of growth, which increase in response to hypoglycemia. Okay, so whenever you have low blood glucose, both of these will increase. They will inhibit cellular utilization of glucose because they want glucose to be in the blood. So they don't want glucose to be taken up by cells. So basically, oppose the effect of insulin, and at the same time, they'll promote fat utilization. So they're basically doing what insulin is trying to not do, which is insulin wants to decrease blood glucose, okay, and it wants to uptake glucose for that to happen. Growth hormone and cortisol don't want that to happen, and at the same time, they want to promote fat utilization. So they'll break down fats, fatty acids. Those will be utilized as substrate. Okay. These effects develop slowly, however, and require hours for maximal effect. Okay. This is important. Remember this. I'm pretty sure we had a question on this in the examination, and we thought it was from a growth hormone lecture, or we thought it was from the ACTH lecture, but turns out it's from this lecture. So you remember this, okay? How do growth hormone and cortisol um, respond to hypoglycemia? They increase gluconeogenesis. Okay. Um, why is this important? Because it's in comparison to glucagon, which will cause glycogenolysis. We'll see that in a second. Okay. Epinephrine, okay, which is one of the adrenal gland hormones, okay, it will increase plasma glucose levels. Again, it's going to be released in response to hypoglycemia. It will increase plasma glucose levels by increasing glycogenolysis in the liver, very similar to glucagon. One difference between epinephrine and the other two hormones that we mentioned is it also activates hormone sensitive lipase, okay? And this will cause the mobilization of fats. Now, one thing that I need to mention for insulin, of course, it's going to deactivate. It does not want hormone sensitive lipase to act because this will cause fatty acids to be mobilized. And we said we don't want fats to be mobilized um, by insulin. All right. Conclusion, enhancement of fat. Okay, so utilization of fat and enhancement of fat is greater with epinephrine than enhancement of glucose. Thus, epinephrine enhances utilization of fat in stressful states. For example, when exercising, when you have a circulatory shock or anxiety, okay, you use up fats more than glucose. Okay, so you'll notice these people, they usually, tend to lose weight. Okay, that's just one of the symptoms. One more thing that you mentioned, which is important in terms of like clinical presentation, if they give a case to you, is a person with hypoglycemia might also sweat and have palpitations, okay, which is mentioned here. Why is that? Because of epinephrine, because that's the type. Okay. The reason for that is epinephrine because it's, again, don't forget its function as an adrenergic hormone. So it's sympathetic, okay? So it's going to cause sweating and you're going to have palpitations, okay? Yeah, this is just to show that it has an effect on glucose, okay? So you need glucose to increase. And again, to relate this to our previous one where we said acetylcholine increases insulin secretion because you want to rest, you want to store. Okay, here, norepinephrine, epinephrine, these are fight or flight hormones. Okay, so for example, exercising, circulatory shock, anxiety, these are pretty stressful states. Okay, so you don't want to just store glucose, etc. You want it to be sent out, and especially for an organ, for example, the brain, okay, which uses only glucose for energy. If you have more glucose, it's better for it, right? So yeah, you want to increase your glucose levels as much as possible. Okay. Here's a question. Large dose of insulin is administered intravenously to a patient. Which of the following sets of hormonal states changes is most likely to occur in the plasma in response to insulin injection? So I'll give you a minute from now. Okay. But at 30 seconds, I'll give you a hint. So it's been 30 seconds. So the hint is that a large dose of insulin was administered. So you have to think about the effect that insulin has and if these hormones have any effect in response to that effect. Okay, time's up. Okay, so the answer is C. Okay, and the reason for that is insulin will cause hypoglycemia, right? Because when you administer insulin, it's going to cause glucose uptake. It's going to do all of its anabolic effects. Okay, in response to hypoglycemia, we said growth hormones released, although it's slow, but it is released. Okay, glucagon. I mean, I didn't mention it, but I did say hypoglycemia. Hypoglycemia is a trigger for glucagon release, and epinephrine 100%, right? All three of these. If I put cortisol in, that will be a correct answer. Okay, so all four of these will respond to hypoglycemia. Okay, but growth hormone and cortisol, they take time to respond. Glucagon and epinephrine, they're pretty quick. Okay, so within minutes, you have a huge response of glucagon, which I showed you right now. Okay, so glucagon is secreted by, if you remember, we mentioned it earlier in histology, is the alpha cells has a function which is opposed to insulin. So whatever insulin does, glucagon wants to do the opposite things. It is primarily a hyper or hypoglycemic hormone. Do you want to increase or decrease blood glucose levels with glucagon? It's going to increase blood glucose levels, right? Because we already have a hypoglycemic hormone, something that wants to decrease the blood glucose levels. So that's the insulin, right? Has a molecular weight of 3,485 to 485 and 29 amino acids. Remember the number of amino acids if you want to. 3485. The reason I mentioned the molecular weight is if you remember insulin, I said 5805. So it's a big molecule, but insulin is a bigger molecule. Glucagon can increase blood glucose concentration by about 25% in 20 minutes, which is a huge increase. Okay, so in cases of hypoglycemia, glucagon can easily make up for it. Okay, yeah, there's nothing else to mention here. Okay, the most important or dramatic effect of glucagon is the ability to cause glycogenolysis in the liver within minutes. Okay, so sure, there's gluconeogenesis and stuff, but like glycogenolysis is really important for glucagon. Pretty sure there's a question on this. Okay, so please make sure to remember this, okay? Glycogenolysis, glycogen breakdown, okay? In the liver. So also remember the organs, hormones, or all of that stuff because there's some confusion between okay, whether this happens in the liver, whether this happens in muscles, what happens in the brain. Okay, so try to differentiate. Glucagon can act on receptors retrieved the following in the liver. So gluconeogenesis. Again, I told you I'll go over the terms really quickly. So glucose, new, genesis, generation, glycogenolysis, so glycogen breakdown. Okay. All right. I promise you this picture was way clearer when I added it. But okay. Major effects of glucagon and effects on blood glucose levels. So we did this for insulin, and you remember insulin was much longer. Okay. Because it's the only hormone that's here. Glucagon, you have other hormones that are helping it out. So you have increased glycogenolysis, increased gluconeogenesis. Both of them are increasing glucose levels. Increases lipolysis. So you're doing what insulin does not want to do. So you're breaking down lipids, okay? You increase keto acid formation. That's called ketoacidosis. If it shows up in urine, that's ketoaciduria, okay? Effects on blood levels, whatever is here. So increases blood glucose, both of these. Increases blood free fatty acids, okay? And here increases blood keto acids. Okay, how does glucagon act? Okay, so I know there's a lot of enzymes, but it should make sense. I'll try my best. Glucagon binds to its receptor, okay? It's a GS receptor. So again, cell signaling is a good lecture to study with this one. GS, if you remember, is G stimulatory. So that's CAMP, okay? Cyclic cyclic adenosine monophosphate. Okay, so that's mentioned here. and adenylate cyclase is used to form this gap. Okay, this is a secondary messenger which will go and activate protein kinase regulator protein. Okay, how do you remember what the next one is? What is it regulating? Protein kinase. So our next substrate will be protein kinase. What does a kinase do? It phosphorylates. Okay, so it adds a phosphate. So phosphorylase B kinase will be activated. What does this? It's again another kinase. So what will this kinase do? It'll phosphorylate something else. It's gonna phosphorylate phosphorylase B into phosphorylase A. Okay. This promotes glycogen degradation to glucose 1-phosphate. Okay. So you're gonna break down glycogen to glucose 1-phosphate, and this is dephosphorylated glucose is released from the cells. That glucose can now circulate in the blood. So now you have you have opposed effect to hypoglycemia, some level of hypoglycemia. Yeah. All right, and this is just showing you that initial process. Okay, so glucagon comes, it binds. So you remember GCPR, the 7 transmembrane protein. Okay, you have this subunit alpha, beta, and gamma. Okay, the GTP replaces GDP. Okay, then this alpha subunit dissociates. This subunit goes, binds this adenyl cyclase. Okay, when it binds to adenylated cyclase, ATP is converted to CAMP. This CAMP will activate protein kinase A activated. Now this kinase will phosphorylate a bunch of other stuff, which we mentioned earlier on, and then glycogenolysis and ureogenesis will occur. Okay, this occurs in hepatocytes, which are liver cells. Okay, glucagon. Its source is alpha cells of the pancreas. Its function promotes glycogenolysis, so breakdown of glycogen, gluconeogenesis, which is formation of new glucose, lipolysis, which is breakdown of lipids, and ketone production, which was from the fatty acid oxidation and stuff. It elevates blood sugar levels to maintain homeostasis. So homeostasis, you remember, well-being of the body when concentration of blood glucose level falls too low. For example, in a fasting state. So in this month, like in the recent days, you're fasting, okay? Your blood glucose concentration levels, they go pretty low, okay? So glucagon's pretty important to like not let you have a hypoglycemic shock, basically, okay? And the regulation, it's secreted in response to hypoglycemia, okay? So whenever there's hypoglycemia, this is secreted. Now, if you wanted to do it the way I told you, like, try to link it. I see hypoglycemia. What else is released in response to hypoglycemia? Growth hormone, cortisol, and epinephrine. What was specific about epinephrine? It also activates hormone-sensitive lipase. What did I say about insulin? It deactivates hormone-sensitive lipase. So, like, try to connect it to other, concepts that you've studied before, so that it sticks with you better. Okay. Okay, so we said insulin is stimulated by glucagon, right? But glucagon is inhibited by insulin. So it's like it gains the benefit by glucagon, but it doesn't help it back. Instead, it inhibits it. Okay. Hyperglycemia. So whenever you have, oh yeah. When you have hyperglycemia, then you don't want more glucagon to be secreted, and somatostatin will also be involved in regulation. Okay. This is a um, pretty big table, and he stressed on its importance. So you need to know receptors, you need to know the signaling pathway, metabolic effects, synthesis, action, all of that stuff is important. Okay, mention this in your recording, so you should know this well. Okay, so glucagon will cause glycogenolysis in the liver. And we said this was one of the most important dramatic effects. It also causes increased gluconeogenesis. So it forms glucose through both of these ways. Its receptor is GPCR, which is a G protein coupled receptor, and GS, which was CAMP, right? Excuse me. And the signaling pathway is adenylate cyclase, CAMP, which is also shared with epinephrine. So epinephrine's metabolic effects are it increases glycogenolysis. See, again, this was what I was talking about. So liver and muscle, okay? So the For example, you can ask you a question, which of the following increases glycogenolysis in the liver and muscle? Okay, and then you can put epinephrine and insulin and a couple of others, and maybe glucagon in as well if he wants to, put in a tricky question. And a lot of people choose glucagon because I said glycogenolysis is a dramatic effect of glucagon. Plus, like the sites are also important. So liver and muscle, okay, and also gluconeogenesis. Um, and receptor is the same, GS, GPCR. However, remember that this is an adrenal gland hormone, so it's going to act on a specific thing called beta adrenergic. And we said beta adrenergic will stimulate insulin. Okay, signaling pathway is adenylate cyclase, CAMP. Okay, again, CAMP secondary messenger, it'll do the protein kinase thing. Insulin will cause increased glucose uptake because you want to decrease blood glucose. You want to cause hypoglycemia. This will be in the liver, in the muscle, and in the fat. So in the liver, you're forming glycogen. In the muscle, you're forming, you're storing protein and stuff. And I think it's better to say adipose tissue, but fat, sure, you're storing fat. Okay. Increased glycogen synthesis. So you're synthesizing glycogen. This will be in the liver and muscle again. So that's where our glucose was, right? There was glucose and there was amino acids. The amino acids were for protein, okay? And then the glucose as glycogen. And decreased gluconeogenesis. So you want to decrease what these two are doing, basically. So you want to decrease blood glucose levels. The receptor for insulin, again, tyrosine kinase, okay? And the signaling pathway is PI3K or phosphoinositide 3 kinase pathway. Okay, so please go over this table really well. Make sure you remember all of this stuff. Most of this is like, you can understand it. Like this whole column, it's understandable if like understood the previous stuff. The receptor for both of these is similar, just like for epinephrine, you need to remember the adrenergic part. For insulin, you need to remember that the receptor is tyrosine kinase. Okay, the signaling pathway is the same for glucagon and epinephrine because it's easy to remember because they're doing the same thing. They're both increasing glucose. Okay, insulin is PI3K. Okay, um, one way you can remember this is I think PI3K was involved with calcium. Okay, so you when you're involving calcium with it, okay, calcium like is uh, very involved with like vesicular movement. So the vesicles containing the glute for transverse, like as a transmembrane thing, will go to the cell surface membrane, they'll fuse with it, and those will allow glucose to be taken up, right? All right, here's a question. Okay, so which of the following stimulates the secretion of both insulin and glucagon from the pancreas? A protein shake recommended for a bodybuilder. Epinephrine injection to treat a severe allergic reaction. Deep fried food available at the county fair. Hyperglycemia due to corticosteroid medication. Hypoglycemia due to prolonged starvation. Okay, I'll give you, yeah, a minute from now. Sure. Okay, that's good enough. Okay, so if you said uh, A, then you're correct. Yeah. Yes. Okay, so why A? Okay, so we said protein shake recommended for a bodybuilder. So what we said for insulin was insulin would, uh, okay, forget the protein part. Besides, it's going to be degraded by the proteases by the pancreas anyway. So you're going to have minus that are going to be taken up. Okay, those amino acids are going to trigger both insulin and glucagon because remember I said for glucagon you have gluconeogenesis, which requires amino acids. And then insulin, I told you it's an anabolic hormone. Whenever it sees amino acids, it's going to want to, put them in a protein and it'll put them in a protein area, so mostly muscles. So that's how you get muscle bulk. Deep fried food, all of the others, not really. Epinephrine injection to treat a severe allergic reaction, probably not. Like the others, yeah, they don't work. A is the correct answer, okay? Because D and E, anyways, like you'll have hyperglycemia. You'll stimulate insulin, but you won't want glucagon. Hypoglycemia, you won't want insulin. You want glucagon. So D and E are out already. So it's between these, and you think about it. Okay, so physiological defense against hypoglycemia. Okay, so you have a decrease in blood glucose level, right? So you're looking at insulin, glucagon, epinephrine, cortisol, and growth hormone, right? Because I said all five of these actually are involved in glucose levels, but only four of them want to increase it, and one of them wants to decrease it. Okay, let's say someone's blood glucose level is decreasing. So what happens with all these hormones? Okay, except for insulin, all of these will increase because we said hypoglycemia will stimulate all of these. This is lowest, which makes sense because I don't know if the order is supposed to matter here, but this one takes the longest, right? You said cortisol and growth hormone, they take the longest. Okay, epinephrine and glucagon in a matter of minutes. And insulin, you want to decrease it because this is already causing hypoglycemia. Okay, so there are four categories here, and one of them has three arrows pointing towards it. So it's like pretty important. Okay, so try to correlate it. So insulin, it'll decrease. If you decrease insulin, you'll have decreased peripheral glucose utilization, which makes sense. You're no longer binding to tyrosine kinase receptors. You no longer have the GLUT4 to like take in the glucose. So you don't longer have the glucose uptake, so you don't have the utilization. What else decrease in insulin does is now you don't have an effect to oppose gluconeogenesis and glycogenolysis. So now this will increase, okay? There are two other hormones that I said will increase this. Those are glucagon and epinephrine. Glucagon mainly wants to increase glycogenolysis. Epinephrine also wants to increase glycogenolysis. I also said epinephrine activates hormone-sensitive lipase, so it's going to want to mobilize gluconeogenic substances, for example, substrates. So substrates could be not like glucose itself, be one from glucose. So substrates could be like, for example, fatty acids, etc. So you want to mobilize fats. And you do that by the hormone sensitive lipase. Cortisol and growth hormone, the reason they take this long is because they want to alter transcription of many genes to conserve glucose. So yeah. Okay. So I met one of my friends first year a couple of days back. I told them about the spell session. They were like, "You put too many memes." So I said, "All right, what do you think of brain rot?" Because I saw this on Instagram, like relevant to my path, and I searched for the other diabetes one. Okay, so here are just two videos. Okay, they're like I think a minute or two long, and I know they're like funny and they're cats and stuff, but like they are a good representation of what really happens. So I hope they help. So watch them when you have time. Okay. So what is diabetes mellitus? Okay. Diabetes mellitus involves impaired carbohydrate, fat, and protein metabolism caused by either. Okay, so all of this will make sense when we discuss the rest of this. So lack of insulin. Okay, this lack of insulin. Okay, is called type 1 diabetes or insulin dependent diabetes mellitus. So the patient is dependent on insulin. Patient with type 1 diabetes is dependent on insulin to like maintain their blood glucose levels. And it's insulin dependent if less than 20% of beta cells are active. You mentioned this like twice, so I think this may be more. It's better to know, okay? And or you could have decreased sensitivity of tissues to insulin. That would be type 2 diabetes mellitus or non-insulin dependent. The problem here is not insulin synthesis itself. You don't have a lack of insulin. You have decreased sensitivity to insulin. So it's not really a problem with insulin itself. It's a problem with sensing insulin, okay? So if you have a problem with insulin itself, you have a problem with all of the anabolic properties of insulin. So you have impaired carbohydrate metabolism. You can't form glycogens. You can't form like fats and adipose tissue. You can have the protein formation in muscles, for example. Okay, I mean you do have fats. We'll discuss this with type 2 because type 2 is a special case because these two are different. Okay, in some ways. So the effects of insulin or lack of insulin, or its of lack of insulin or its sensitivity will mean inefficient uptake into all tissues and organs, etc. Except so let's say you have a problem with insulin. So like it should not normally affect your entire body, but it doesn't affect something. What is it? Something that is not dependent on insulin. Like I will give 5 seconds. But if you said um, but if you said brain, uh, yeah, okay, there we go. But if you said brain, then you're absolutely correct. Yeah, good. So this is really important. Okay, the brain does not depend on the utilization of insulin or the blood glucose levels or whatever. Okay, it's just permeable to glucose, and it because it only uses glucose for energy, it's not supposed to be limited by a specific hormone. So yeah, it makes sense. So because of this problem in diabetes, blood concentration of glucose will increase. So you'll have hyperglycemia, and you'll also have cellular utilization of fats and proteins because now there's nothing that's stopping the proteins from being broken down. There's nothing that's stopping lipolysis from occurring. Okay. Okay, so according to your slides, this slide is supposed to be like 2 slides later, and you'll have the first aid ones earlier on. But I want to mention this right now because the symptoms then make much more sense in this order, I think. So let's say you have a problem with insulin deficiency or severe insulin insensitivity. So you have insulin resistance, basically, which is type 2 diabetes. This one's type 1 diabetes. So first off, insulin's supposed to increase glucose uptake. So now you don't have insulin, you decrease glucose uptake. Insulin's supposed to decrease this, now it's increased. So now you have more breakdown of glycogen. You'll have more reduction of glucose. So now you'll have hyperglycemic states, right? Which is mentioned here. You have hyperglycemia, and glycosuria is basically glucose in the urine. So recall renal. Okay, I know there's like a lot of links to this lecture, but it's really important. Recall renal. So above a certain threshold for glucose, okay, you'll have glucose in the urine. Okay, and that threshold's crossed with diabetes. So now you'll have glucose in urine. You're not supposed to normally have it because it's reabsorbed with, I think, sodium. Yeah. Now, if you have glucose freely floating in blood, now you have hyperglycemia, okay? Glucose doesn't have specific transporters to take it into cells because those transporters were triggered by insulin, which we don't have, okay? So now that glucose is going to stay in the blood. What can come out of the cells is water, okay? It's because of the osmolarity, osmolar pressure and stuff. You've taken this in renal, you've taken this in FND, okay? So now, since you have increased plasma osmolality, all the water is going to come out. Now you're going to have thirst, okay? The medical term for thirst is called polydipsia. Osmotic diuresis is basically, you have the water coming out, and all of this water and the glucose, it's going to be secreted with the urine. You have loss of water. You have loss of sodium because I said sodium ion. Okay, I didn't mention this because we didn't have a separate slide for it, but sodium ion retention is also dependent on insulin, okay? So it does have an effect on kidneys, and one of the effects on kidneys is sodium ion retention. You also have loss of potassium, and that makes sense. We said insulin will cause increased permeability to potassium. Okay, insulin causes the uptake of glucose with potassium. Okay, insulin can't cause an uptake of glucose because it's not even there. Okay, so potassium won't be taken up. So potassium stays in blood. So the potassium leaves with glucose, and yeah, you have loss of all of this. All of this loss will cause hypovolemia. Hypovolemia means less blood volume, basically. Okay, so now you'll have circulation failure. Okay, and you'll take this in PLD, I think, again with Dr. Rehan. Okay, he's going to explain to you how hypovolemia affects circulation failure, but it makes sense. Okay, if you have less blood, you have less tissue perfusion because it's now you don't have enough blood to go to all the different organs, different areas. Okay, so let's say like distal areas or areas further away from the center of your body. So for example, the hands and the feet. Okay, maybe they don't receive all the blood that they're supposed to. Okay, when you don't have proper tissue perfusion, then what happens? What does happen? What happens then? Okay, you have anaerobic respiration, right? So pyruvic acid will be converted to lactic acid. So you have increased serum lactate. This will cause an anion gap and metabolic acidosis. Okay, I'll get back to these in a second. Okay, let's go back up here. Okay, let's go here. Okay, protein breakdown. Okay, now insulin was supposed to inhibit this. Insulin's not there. Proteins won't be. The proteins just break down. Okay, there's less proteins. There's weight loss. Sure. Okay, specifically muscle wasting. So muscle bulk is lost. Okay, and those amino acids, okay, that are formed from the protein, now they can be used for gluconeogenesis. This is why the protein breakdown happened in the first place. Lipolysis, okay, so you break down fats, okay, increased plasma free fatty acids, okay, you'll have increased ketogenesis, ketonemia, which is ketone bodies in blood, ketonuria, which is ketone bodies in the urine. These will cause vomiting, okay. Like I said, a feature about ketones is they have a fruity smelling breath. The patients will present with a sweet smell, sweet smelling breath. Okay. Now, ketones in blood and this serum lactate. Okay. Keto, we call this ketoacidosis. Okay. Serum lactate, lactic acid. That's also acidosis. You have metabolic acidosis here. Acidosis, which means hydrogen ions. So full circle. Okay. Go back to my last session on controlled breathing if you want to. Hydrogen ions can cause hyperventilation. And for specifically for diabetes, they call it osmolar respiration. Okay, so that is this entire chart here. Okay, if you have any problems with this chart, just feel free to reach out. Yeah, the circulation failure will cause coma death. Okay, okay. Diabetes mellitus. Okay, so acute manifestations include polydipsia, which you said was thirst. Okay, polyuria, so the patient wants to urinate more. I mentioned like the main terminologies are in the notes section of these slides, but the rest I didn't really have time to make slides on, so I'm going to explain them right now, of course, all of this. Okay, but if you have any specific questions, please do ask. I'll try my best to, fix up something if I can later on. Polyphagia, so it's related to satiety and stuff, okay? The reason this is important, these three are important. Sure, all of the rest of this is important too, but these are very important for a clinical case scenario. So these are usually seen with diabetes type 1, as we'll see in this, the comparison chart between type 1 and type 2 in a second, okay? There's weight loss, okay? Why weight loss? Because you're breaking down proteins, you're breaking down, you're causing lipolysis, okay? Diabetic ketoacidosis, which is a severe manifestation of type 1 diabetes mellitus, okay? Because you have ketones in the blood, okay? That's the osmolar respiration and everything. Hyperosmolar hyperglycemic state, this is more associated with type 2, okay? Rarely can be caused by unopposed secretion of growth hormone and epinephrine. Okay, so also seen in patients with glucocorticoid therapy, steroid diabetes. Okay, um, yeah, okay. Because unopposed secretion of growth hormone and epinephrine, you understand that, right? Because like there's hyperglycemia and stuff too. Okay, chronic complications. So you can have non-enzymatic glycation. There's two things that you need to understand here: small vessel disease and large vessel atherosclerosis. Okay. Small vessel disease. So all areas of small vessels, okay, they're affected by this diabetes. Okay, so there's basically diffuse thickening of the basement membrane. And one of the important things that should come to your mind right now, you should try to relate this to renal. Okay, so glomerular problems. Okay, because you have nephropathies and stuff. Because nephrons, they have the glomeruli, they have these small capillaries. All of these small vessels are around those areas, right? Because that's how you reabsorb all the sodium, the glucose, the potassium, all that we mentioned earlier on, everything that you took in renal, right? Um, but one of the more important ones, which will take again second year, second semester, is retinopathy. Okay, so the retina again, that's related to the eye. Just know this. Okay, it's related to the eye. There's very small vessels here. There's hemorrhaging. Okay, exudates, microaneurysms, which is like basically blood vessels burst. Okay, really small blood vessels burst. That's called the microaneurysm. Vessel proliferation. Any of these things happen, they cause an increase in pressure behind the eye. This is called glaucoma. Okay, neuropathy, which is mentioned again later on here. Okay, I'll get to that in a second. Nephropathy, like I mentioned, with the kidney problems, progressive proteinuria, so proteins are found in urine. Okay, so initially microalbuminuria, so small albumin is one of the proteins. Okay, in urine, ACE inhibitors are neuroprotective and arteriosclerosis. Okay, so we're going to talk about the atherosclerosis for a second, but hypertension and both lead to chronic renal failure. So basically, your eyes can be affected. So that's one. And the major part of the rest of this is that the small vessel disease will affect the kidneys, okay? Specifically the nephron region. Yeah. Large vessel atherosclerosis. Okay, so you have coronary artery disease, peripheral vascular occlusive disease, gangrene, okay? Gangrene is basically um, necrosis. Wait, that's way too. Uh, like, how do I explain this? Okay, uh, think about it like this. Basically, there's loss of blood supply to your distal, like for example, fingers and stuff. Okay, I guess frostbite is one way to think about it. Something like that. Okay, so there's loss of blood supply to your fingers, etc. Okay, eventually your tissues will die out because they're not receiving any blood. They're not receiving any oxygen. Your cells die, which is in the process called necrosis. Okay, and if this happens at a wide scale, that's basically called gangrene. Okay, gangrene is like a coagulative necrosis, which will take in POD second year. Don't worry about it. Just remember that will cause limb loss, cerebrovascular disease. Cerebral is cerebrum, so that's the brain. That's the block I'm taking right now. Vascular is related to blood vessels. Okay, so again, you'll have problems with blood vessels in the brain. Okay, so you can have any sort of like how do I put this? Any sort of like mental issues, maybe like some sort of cognitive problem, something with the brain. Myocardial infarction is the most common cause of death. So this is something you can remember. Okay, this is something important. Atherosclerosis because there's more cholesterol. Okay, because you're causing lipolysis and stuff because you don't have the insulin to regulate the fats. Okay, osmotic damage. Sorbitol is something that's just formed due to the accumulation of glucose. So whenever there's hyperglycemia, you have sorbitol formed. And accumulation organs with aldose reductase and a decrease or absence of sorbitol dehydrogenase. So you'll have neuropathy. Okay. You take this in neuro. Okay. So the block I'm taking right now, we'll take this in detail. There's something called distal axonopathy right here. Um, but for you to understand, for as a first year, just think about it like this. So hands and feet are affected basically because you're just remember it like this. Yeah, you have sensory loss, so you lose sensation. You can't feel stuff in your hands and feet. Okay, so how this patient will present to you is because they can't feel anything. They'll basically bump their feet into like tables and stuff. So they'll have foot deformities, scars and stuff. Okay, because they seriously can't tell there's a problem there. Okay. So motor problems, which is with movement sensory, it's called a glove and stocking distribution, very characteristic term for diabetic neuropathy. Okay, and autonomic degeneration, which is like sympathetic parasympathetic. Okay, cataracts is like the lenses of the eye. It's relevant to the lens of the eye when you're in pain. Yeah. Okay, that's it for this one then. Okay, type 1 versus type 2 diabetes mellitus. Okay. So this is important. You need to be able to compare it. Okay. So start with the primary defect that 1 degree stands for primary. Okay. So what's the problem in type 1 diabetes? You have an autoimmune destruction of beta cells, for example, due to glutamic acid decarbonase. Don't worry about this too much. I mean, sure, remember the name, you have 3 weeks, it's fine, okay? But like the problem is antibodies, autoantibodies. So you'll study this, it's like in your immunology, but you have autoantibodies. Basically, your body can't like tell that it's attacking you. It just attacks you as if it's like as if you are the foreign problem, you know? You are like another bacteria or a virus. So it attacks the cells of the pancreas. So it attacks the beta cells specifically. And we said the beta cells secrete insulin. If you have autoimmune destruction of beta cells, you took, I think, antigens and stuff with Dr. Garland. So assume that the beta cells have antigens, okay? The autoimmune cells come, they attack these antigens, they destroy these beta cells. Now you can't produce insulin anymore. So that's why you have a lack of insulin, okay? So remember that type 1 has a lack of insulin because it's going to come throughout the stable again and again, okay? In type 2, you have resistance. You don't have a problem with producing insulin, okay? But eventually, so what happens is you have resistance to insulin. You're secreting insulin, okay? But it can't do its job to like the effect it's supposed to. So what's going to happen is you still have hyperglycemia. Now, your beta cells are still going to receive that glucose in the proportional rate that we mentioned earlier to the GLUT2 transporters, and it's going to still feel like there's hyperglycemia. So it's still going to produce more and more insulin, okay? And eventually it's going to burn out the cells. That's called progressive pancreatic beta cell failure. Okay. Eventually, for patients with this, okay, in a chronic state, you're going to use sulfonylureas, all of the other drugs that we mentioned, like all the other treatments, because insulin, you can't depend on your beta cell insulin anymore. Insulin necessary in treatment, always in type 1, because that's the main problem here, okay? And that's why it's also really sensitive to insulin, which is mentioned here, okay? Because, yeah, you can produce insulin because your beta cells are destroyed. So if you give insulin, you fix the problem that's present here. Here, your problem is with the receptors, because not exactly the receptors, but it's like sensing insulin. So it's not something that's like really easy to fix. But like later on, like you do get insulin with sulfonylureas, the treatment, yeah, because I said sulfonylureas, they bind and block the potassium ion channels, the ATP sensitive potassium ion channels, and that causes the depolarization that will cause the influx through the calcium ion voltage gated channels of the calcium ions. Okay, ages. So for type 1, less than 30 years, yeah, they'll be affected. Type 2, more than 40 years. So you can expect a younger patient. So what he can do is he'll give you a case. A 16-year-old child comes and he has some problems with diabetes. So I'm not saying like always assume it's type 1 diabetes, but like it's most likely type 1. Okay, so that's what your thought process should be there. Okay, type 2, over 40 years. So he'll bring like a 65-year-old man comes in with a problem. Yada, yada, yada. Okay. Association with obesity. Do you have obesity in type 1? No. Why? Because you have no insulin, right? So now you can have lipolysis unopposed because insulin stopped the lipid degradation. Now you can have lipolysis to maybe, like for example, epinephrine. Okay, hormone sensitive lipase is activated. You have lipolysis. You have ketone bodies and all of that. So you have diabetic ketoacidosis later on as well. But it's like you won't have obesity. You'll have weight loss actually here. Okay, here, yeah, because you do have insulin. Problem is with sensitivity, but to some extent, you're still sensitive to insulin in some parts, so you'll not like, so you won't have lipolysis in those parts. So you will have weight gain. Okay. Genetic predisposition, relatively weak in type 1, but it is high in type 2. So for type 2, so for example, a 65-year-old guy comes in, you should expect that someone in their family probably also has diabetes. So it has some relation. It's not like just randomly the person loses, sensitivity to insulin, that some genetic predisposition is there. Over here, since it's an autoimmune destruction, maybe it's because of an infection and then your body reacts in an abnormal way. And that's the autoimmune destruction. So it doesn't have to be genetic, but it could be. You know. Okay, association with the HLA system. Yes, HLA DR4 and DR3. We'll take this later. Don't worry. Okay. HLA stands for human leukocyte antigen. Okay. 4 and 3, just remember it the way first I put it. So these are good ways to remember. Okay. So 4 minus 3 is type 1. Okay. So it's he asks you which, and I think he did ask this, which HLAs are associated with diabetes type 1, and he can give you 1 and 2, 1 and 3, 2 and 4, uh, 2 and 3, and then 4 and 3. Okay, so 4 and 3 will be the correct answer. Okay, type 2, no. Okay, glucose intolerance is severe in type 1 because you have no insulin. Here, mild to moderate because you do have insulin. It's a problem with sensitivity, but it does act on some parts, so it's fine. Insulin sensitivity is high here. Here, that is the problem. So that's why it's low. Here, it's high because you don't have insulin. So if you do give insulin, it's going to act like really well. It's a very good treatment, okay? But there are other things you're going to add to the treatment. I'm going to show you what that is. Ketoacidosis is common here, but rare here. Why is it common here? Lipolysis, okay? Beta cell number in islets are low. Why? Because that's the problem, primary problem here. That's the destruction of the beta cells, the autoimmune destruction. Your white blood cells came, they destroyed the beta cells. That's why they're low here. Here, it's variable because eventually you have beta cell failure because it's like burnt out and stuff. So initially not hurt, but like later on maybe. Serum insulin levels very low because again, it's not producing it. Here, variable again. So it could be pretty high because hyperinsulinemia, but there's still no sensitivity. Classic symptoms of polyuria, polydipsia, polyphagia, weight loss. Again, polyuria, wants to urinate more. Polydipsia, thirsty. Polyphagia related to satiety. And weight loss because of lipolysis. Because of lipolysis. So because of lipolysis, this is common in type 1. Type 2, we said not enough lipolysis, so sometimes, okay? And histological features, if you look under a microscope, you'll see an islet leukocytic infiltrate, which makes sense because you have autoimmune destruction. So what causes the autoimmune destruction? It's the leukocytes, which is, the white blood cells and stuff. Here you'll have amyloid polypeptide deposits. Don't worry too much about it, just memorize it the way it is, okay? All right. How do you test for diabetes? There are 3 tests, okay? One is hemoglobin A1C, okay? This one is called glycated hemoglobin. If it's more than or equal to, and you mentioned this in your class, there's like differences now, just like, sure, stay with the slides, okay? Stick with the slides. Or maybe Yahya's team, the notes committee, did the transcript, like go over the diagnostic cutoffs that Dr. King gave, okay? Um, yeah. So what they do is they check for hemoglobin A1C, and what this gives an indication of is the blood glucose level over the past 3 months. So this is, a bit vague, I guess, right? Because like it's too big of a time period. Okay, but still remember it because it's important. Okay, it's very important for diabetes specifically. Fasting plasma glucose, if it's more than or equal to 126 milligram per deciliter after fasting for more than 8 hours, it's a sign that, okay, there is hyperglycemia. Okay, so yeah, but the most important one that he mentioned, the most important test for diabetes is 2-hour oral glucose tolerance test. So what they do, it should be greater than or equal to 200 milligrams per deciliter. So what they do is they measure the patient's blood glucose. Okay, fasting blood glucose, of course. Excuse me. So fasting blood glucose, of course. Okay, then they make the patient consume 75 grams of glucose in water. Okay, 2 hours after consumption, they measure blood glucose levels again. If they are greater than or equal to 200 milligram per deciliter, that means it's most likely diabetes. And that makes sense because like I said, insulin is going to immediately act to cause hyperglycemia. But if you don't have insulin or your insulin isn't sensitive enough, it won't happen. So you do have diabetes. Your patient has diabetes. All right, diabetic ketoacidosis. Which type of diabetes is this more associated with? Type 1, right? So one of the most severe complications of diabetes, okay, usually due to some non-compliance. So if a patient has been told to take insulin, but they don't do it, okay, or increase insulin requirements from stress. So let's say I'll give you the same example he gave to the class. So the patient is like a 6-year-old child or something, okay, maybe a bit older, okay, and he or she has increased stress due to, for example, an infection. He had a fever or something, okay. Um, so this will cause increased insulin requirements because now you want to have hypoglycemia to like moderate the blood levels. But if you don't have enough insulin, okay, you're not going to have enough um, inhibition of lipolysis to prevent this diabetic ketoacidosis from happening. Okay, which is mentioned here. So excess fat breakdown occurs, that's lipolysis, increased ketogenesis from increased free fatty acids, which are then made into ketone bodies through what? Beta hydroxybutyrate more than acetoacetate. Okay, again, mole, please revise this, okay? Usually occurs in type 1 diabetes as endogenous insulin, and type 2 diabetes usually prevents lipolysis, okay? And that makes sense because in type 2 diabetes, you do have insulin. So insulin, even though it's less sensitive to insulin, it's still going to prevent lipolysis to some extent. But it's like in type 1 diabetes, you don't have insulin at all. So endogenous insulin is so low. And how do you know endogenous insulin is so low? Because of a certain marker called the C-peptide, okay? Type 2 diabetes, C-peptide is going to be pretty high because it's going to be hyperinsulinemia, right? You're checking for endogenous insulin. How do I know my patient's non-compliant with insulin? Again, C-peptide, okay? Signs and symptoms. DKA or diabetic ketoacidosis is deadly, okay? So a lot of these here. So deadly, delirium or delirious, basically delusional psychosis, okay? Customer respirations, like I said, rapid, deep breathing, okay? Because there's acidosis. Abdominal pain, nausea, vomiting due to the ketones like we mentioned earlier, dehydration, and thirst. Why? Because of the plasma osmolarity problems that we mentioned earlier, right? Fruity breath or odor, this was what I was talking about. This is very characteristic. If he gives you this, it's like an easy mark. Okay, he wants you to guess it. So he wants you to get that mark because it's like that one, very easy to tell. This is very characteristic, okay? Even if it's not asked in ERP, it can be asked in more. So please remember this, okay? Fruity breath or odor is associated with ketones. Labs for diabetic ketoacidosis, so hyperglycemia. You'll find that there's hyperglycemia. Hydrogen ions will be high because, like we said, there's acidosis. Higher hydrogen carbonated ions will be low. So there's an anion gap. This is, I think, more relevant to your renal parts. So you have to, yeah, you should know this by now. Metabolic acidosis, just like hydrogen ions are high. Okay, blood ketone levels are high. That's why hydrogen ions are high. There's leukocytosis. Okay, hyperkalemia. Why hyperkalemia? Because you don't have insulin, right? Insulin can't take up potassium. So that's why you also have depleted intracellular potassium ions. And due to the transcellular shift from decreased insulin and acidosis. So osmotic diuresis occurs, which we mentioned earlier. You have increased potassium loss in urine, so your total body loses potassium. Okay. So why is this important? We'll mention this in treatment. Okay. Um, Complications that can occur due to diabetic ketoacidosis, life-threatening mucormycosis. So this is something Dr. Boda, who is a microbiology doctor and doctor in year 2, taught us. Yeah, it's a fungal infection. Don't worry about it. Okay. Just remember that. Okay, yeah, with diabetes you can have fungal infections, etc. Cerebral edema, cardiac arrhythmia, so problems creating a rhythm in the heart, and of course, because of the cardiac arrhythmias, you can have heart failure. Treatment is IV fluids, IV insulin, okay? IV fluids to replete because you have osmotic diuresis, you have the hypovolemia that we mentioned, right? IV insulin to make up for this, okay? Potassium to make up for this. Why do you give, how you give potassium with glucose if necessary to prevent hypoglycemia? So for one, you're giving insulin, so there's going to be hypoglycemia. So you give glucose as well, so there's no hypoglycemia. At the same time, glucose has a second function, which is to prevent which is to allow the uptake of potassium. So, like I said, associate these together, please. Okay. Next, you have hyperosmolar hypoglycemic state. Now, the last one was associated with type 1. This one's with type 2, so stage of profound hyperglycemia induced dehydration and increased serum osmolality, classically seen in elderly type 2 diabetes, elderly above the age of 40, like I mentioned, with limited ability to drink. So hyperglycemia with excessive osmotic diuresis, there's dehydration, okay, because of the osmotic diuresis, and eventual onset of hyperosmolar hyperglycemic state, okay. Symptoms include thirst, which is polydipsia, polyuria, lethargy, okay. Focal neurological deficits, that's basically focal means at a specific location, neurological neurons deficits, and this includes seizures, okay, can progress to coma or death if left untreated. So as you see, there's like two symptoms here which are very similar to the hypoglycemic shock. When you do the labs, you'll find out there's hypoglycemia, often more than 600 milligram per deciliter. This is pretty high. In diabetic ketoacidosis, your hypoglycemia is about 300 to 400. So this is way higher, okay? Increased serum osmolality, okay, because of the hypoglycemia. So more than 320 osmolar per kilogram. No acidosis because you still have insulin. It's the problem with sensitivity. It's not the problem with producing insulin or inhibiting the lipolysis. So there will be no ketoacidosis, okay? Like it's mentioned here, so ketone production is inhibited by presence of insulin. And treatment is aggressive IV fluids and insulin therapy, okay? So you give IV fluids to replace the fluids lost by osmotic diuresis and insulin therapy so that you fix the symptoms with the hyperglycemia, right? Glucagonoma, okay? You'll learn later on that OMA is usually supposed to be for benign tumors, okay? So a tumor of pancreatic alpha cells, because it's glucagon, overproduction of glucagon presents with dermatitis, necrolytic migratory. Why did they give such difficult things? Okay, dermatitis is basically uh, inflammation, okay, and derma is related to skin, okay. So take this in MSI. Diabetes, hyperglycemia, okay. DVT is deep vein thrombosis. You'll take this in POD, but basically it means thrombus is basically like a blood clot, okay? Deep vein, blood clot inside, deeper veins. That's basically it. Declining weight, okay? Um, Depression, and treatment is octreotide, okay? Octreotide is an analog or basically mimetic, something that's very similar to somatostatin, and surgery to resect the tumor. So like remove the tumor basically. Insulinoma. So this one was a tumor of the alpha cells. If you have a tumor of the pancreatic beta cells, you have an overproduction of insulin. So whatever you see with insulin, just amplify that. And that's what you see here. In diabetes, we reduce whatever we saw with insulin so that we saw the symptoms. Okay, here we're amplifying it. Okay, so we're increasing it basically. So you'll have hypoglycemia. You will see Whipple strike. This is very important, very high yield. Okay, he doesn't spend too much time on it, but he can ask about it, and it's really important. Low blood glucose. We know why. The hypoglycemia, right? You're not you are taking up blood glucose. Okay, so this time it's normal, right? But normal insulin, but like it's produced in great amounts. So now it's like abnormally, taking up glucose. Symptoms of hypoglycemia, so lethargy, syncope is like problems with, you think it's problems with the heart, and it affects the brain systems as well. So patients may faint and stuff. And diplopia is like double vision and stuff, okay? And resolution of symptoms after normalization of glucose levels. So your symptoms will resolve basically whenever you return the blood glucose levels to normal. Right now, there's hypoglycemia, but when you provide glucose and stuff, which will be one of the treatments, okay, you'll have resolution of symptoms. Symptomatic patients have low blood glucose and increased C-peptide levels. Why increase C-peptide? Because it's always secreted with the same secretory vesicles, okay? So this gives you a measure of the endogenous insulin. Where else will you have increased C-peptide levels? In the diabetes type with insulin resistance, okay? So type 2 diabetes. 10% of cases with MEN 1 syndrome, that's multiple endocrine syndrome, basically, okay? And the treatment is surgical resection. So whenever there's a tumor, surgery is important so that you can remove the tumor, okay? This is the last one, okay? So pathogenesis of diabetic ketoacidosis, insulin non-compliance, or requirements due to stress infection. So we basically discuss this, okay? Ketone bodies will be high because there's lipolysis, okay? Insulin can prevent this from happening. In osmolar hypoglycemic state, you do have insulin, which prevents the lipolysis from happening, so you don't have acidosis, but you do have these serum osmolarity problems because you still have the hyperglycemia. So insulin is present, ketones are absent. Here, insulin is not present, and ketones are present. Okay. The signs and symptoms, again, this is repeated, so he will most likely ask about this. Okay. Again, potassium ion problems because insulin is no longer there, so you can't reuptake it. And then over here, you have hypoglycemia, increased osmolality. The complications, like we said, you can have a fungal infection here. Here, you have coma and death if they're untreated. In both of these, you have the treatment of IV fluids, IV insulin, potassium ions to replete intracellular stores, and glucose may be required to prevent hypoglycemia from insulin therapy. Okay, so that was the last slide. Now I got a couple more questions in there. I'll give you about like uh, 30 seconds. This is easy. Okay, so the answer to this question, let's read it very quickly. A 10-year-old girl with no prior medical history presents to the clinic because she's not feeling well. The mother says that the girl has been very thirsty lately and urinating more frequently. She has lost 4.5 kgs, okay, over the past year. Laboratory studies show that markedly elevated blood glucose level and a decreased insulin level. Levels of a specific antibody are also elevated, and antibodies against which of the following organs are most likely present in this girl. Okay, before I sure tell you the answers, right? But like one of the things that you want to do when you see a case like this is try to identify what you're seeing here. So it's a 10-year-old girl. She's very thirsty lately. That's polydipsia. Polyuria. Okay, she's lost weight loss. All right, markedly elevated blood glucose levels. That's hyperglycemia. Decreased insulin level, and levels of a specific antibody are also elevated. It's probably autoantibodies against their beta cells. Okay, and lastly, the thing that confirms that this is type 1 diabetes mellitus involving the pancreas is the age. The age is less than 30. Okay, next question. It's the last one. 19-year-old man is brought to the emergency department due to weakness and confusion. Family members report that for the last 2 weeks he has experienced polyuria, polydipsia, and increased appetite. Medical history is remarkable for an appendectomy at the age of 12. The patient does not use tobacco, alcohol, or illicit drugs. Family history is significant for hypothyroidism. His brother. Temperature is 36.7 degrees. Blood pressure is 90 per 60. Pulse is 97. Respirations at 22. On physical examination, the patient is lethargic but follows simple commands. His breath has a fruity odor. This patient is most likely deficient in a hormone that normally performs which of the following functions. So I'll give you a minute. Sure. Try to think about the type of diabetes as well. Okay, I think that's good. Yeah, sure. So the answer here is B. Okay, so it's a 19-year-old guy. Okay, so that's again below 30. We know the case scenario, so we know it's diabetes, it's type 1 diabetes. Okay, polyuria, polydipsia, like I said, these are important for case presentations. Or he could ask you, okay, there's a patient with type 1 diabetes, what do we expect to see? And he can give you polyuria, polydipsia, decreased appetite, polyuria, polydipsia, increased appetite, polyuria, something random, poly, and then polyphagia, polyuria, polydipsia, polyphagia. So that would be the correct one, right? So you just need to know the respective, like what each of these means and like the diabetes case that's associated. Okay. The rest of this is like you can skip over most of this and you can still answer. It says breath has a fruity odor. That means ketones. Okay. Even if you just read these last 2 lines. Okay. His breath has a fruity odor, so I'm thinking ketones. This patient is most likely deficient in a hormone which performs receptor functions. If he has ketones, that means this patient is deficient in insulin. Now I just have to look for something that insulin does. We said glucagon increases insulin secretion, but insulin decreases glucagon secretion. Insulin inactivates hormones in the lipase. This is the function of epinephrine. It decreases muscle glucose glycogen synthesis. That's wrong. Okay. Glycogenolysis is a function of the others, right? This increases muscle glycogen synthesis. Insulin, I mean. Increases hepatic glucose production. That would be gluconeogenesis. That's wrong again. And increases in the glucose reabsorption. Wrong. Okay. So that's what's B. So I know it's a long lecture. It's recorded. So if you have questions, feel free to ask them. You have my contact in the um, initial slide. And yeah, that's the end of the lecture. Please do not forget to fill out this survey. Okay. Please scan the QR code. Give your feedback. If you want more memes, it's fine by me. Um, yeah, let me know if you want anything to be changed, or if you want have any questions for anything in particular, please do ask. All right. Thank you so much for watching. Ramadan Kareem, and have